So we've already talked about how Mendel arrived at the first law, the law of segregation, where he determined that the two alleles for a specific trait segregate independently. But now we're going to talk about how he arrived at his second law. So one thing he wanted to decide and figure out was how can we tell the genotype of an individual with a dominant phenotype, because we know that that individual could either be homozygous dominant or they could be heterozygous. So if you're purple flowers, for example, you could be big P, or you could be big P, little p. So the way that he figured this out was he decided to do something called a test cross. And this is where he didn't know the genotype of the parent involved, so he wanted to figure out what that parent's genotype would be. And he would do this by breeding the mystery parent with a homozygous recessive individual. So, for example, a purple flower that he didn't know the genotype with a white flower, because he knows that the white is little p, little p. If any of the offspring display the white flower, then he knows that the parent has to be heterozygous, because if it was homozygous dominant, he would not have gotten any white flowers, just like in the F1 generation. So this is where we don't know what the purple flower could be, because it's purple. It could be either big P, or big P, little p. And we're going to cross with the white flower, which we know is little p, little p. Then we can see what would happen. So if we end up with any white flowers, we know that it has to be a big P, little p parent, because with the big P, little p, we get the option of having a homozygous recessive offspring. If the parent is homozygous dominant, however, that dominant gene is going to be passed to all of the offspring, and we will never see the recessive phenotype in that first generation. All of the offspring will be purple. And so we can go backwards and use the test cross accordingly. So in doing this, Mendel derived the law of segregation by following a single character. And he determined that in that case, the F1 offspring were considered to be monohybrids. This means that they were heterozygous for one character. And when you cross two heterozygotes, you get what's known as a monohybrid cross. However, he decided to see what would happen if he tried to follow two characters or two traits at the same time. And he would cross two true breeding parents who differed in two characters. So, for example, a purple flower with round seeds with a white flower with wrinkly seeds. And he would call these dihybrids, because in the F1 generation, they would be heterozygous for both of those characters. So if he had true breeding parents, for example, capital P, capital R with lowercase p, lowercase r, you're going to get all the offspring will be big P, big R, little r. And this is called a dihybrid cross. In doing this, he was trying to determine whether two characters could be passed on to offspring either together or were they passed on independently. And what he ended up finding, this is an example of the P, so he found that the seeds could be either yellow or green, and they could be round or wrinkled. So the dominant traits are yellow and round, and the recessive traits are green and wrinkled. And when he crossed these, he saw that the entire F1 generation was heterozygous. Then he did a dihybrid cross with two heterozygous F1s, and what he ended up finding was a phenotypic ratio of about 9 to 3 to 3 to 1. So 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 green wrinkled seed. And in doing this, he realized that each of these traits is independently assorted. These traits are not inherited together, they're inherited separately. So this gives us a little bit closer up view. But his dihybrid experiment results demonstrated the law of independent assortment, and he derived this law using his experimental results. This law basically states that each pair of alleles segregates independently of any other pair during gamete formation. So that applies to the genes on the chromosomes that are not homologous or those far apart on the same chromosome. So this basically means that when a new organism is forming, just because a parent donates an allele for a purple flower doesn't mean that it's just because it has an allele for a purple flower, it's going to also send an allele for a yellow seed. It doesn't matter. Everything is independent. So each of the alleles is assorted independently. And this piece is important because this law that Mendel observed only applies to genes that are not homologous and those that are far apart. But we've since found out that genes located near each other on the same chromosome tend to be inherited together. But for Mendel's purposes, and especially considering he didn't know anything about chromosomes, it's pretty amazing that he was able to determine that these alleles are transferred to the offspring independently. And we will stop there. That is all about Mendel and his pea plants.